Now I'd like to introduce today's speaker, Dr. Joanna Tannous. Joanna is an associate staff scientist serving as a fungal geneticist for the Secure Ecosystems Engineering and Design Scientific Focus Area at Oak Ridge National Laboratory. Joanna is working on developing CRISPR systems for the control of fungal plant pathogens associated with Populus. She is also associated with the synthetic biology group at Oak Ridge National Laboratory. She specializes in developing editing tools in non-model fungal species to elucidate the molecular mechanisms and pathways regulating fungal pathogenicity, metabolism, and the interaction of the fungus with its host and surrounding microbial communities. Her research is also focused on the discovery and characterization of novel fungal metabolites through genetic manipulation and omics approaches to study their role in fungal host interactions and microbiome dynamics. Joanna, thanks for participating in this webinar program. We are looking forward to hearing about your work, and at this point, I'm going to turn things over to you.
Hi, everyone. So I'm Joanna Tannous, an associate staff scientist at Oak Ridge National Laboratory. I'm very happy today to be presenting some of the research that I'm conducting in my lab that focuses on developing CRISPR-Cas9 gene-drive system and the plant pathogenic fungus Sphaerulina musiva to mitigate diseases on poplars. So here's the outline of my talk today. Although David presented me, I'll give a brief educational background and experiences that I had during my career so far. Later, I will introduce the Fungal Kingdom for those of you who are not familiar with the Fungal Kingdom. I will give a brief background on the Populus and Sphaerulina musiva pathos system and the latest advancement in disease management of this pathos system. And later, I will get to the overall approach that we are using towards the development of the safeguarding CRISPR-Cas9 gene-drive system in Sphaerulina musiva. So I started my science career with a bachelor degree in life and earth sciences and biochemistry from St. Joseph University of Beirut in Lebanon, where I'm originally from. And from the same university, I got my master's degree in food chemistry in 2010. And in 2015, I got my dual PhD in pathology, toxicology, genetics, and nutrition, and a PhD in chemistry from the University of Toulouse in France and St. Joseph University of Beirut. After my PhD, I completed a 3-year postdoc in the fungal genetic lab of Dr. Nancy Keller at the University of Wisconsin-Madison, followed by 1-year postdoc at the Research Institute of Horticulture and Seeds at the University of Angers in France. And in July 2020, I joined Oak Ridge National Lab as a postdoc in the synthetic biology group, working on the secure engineering and design science focus area, or short SFA projects, before transitioning in December 2022 to become an associate staff scientist at ORNL. Throughout my career, I worked on developing genetic engineering tools to transform model and non-model fungal organisms, some of which have never been genetically transformed before. So I developed those fungal genetic engineering tools for different applications. I use them to either answer questions related to fungal metabolism, so either to characterize novel fungal metabolites by overexpressing certain genes in their putative biosynthetic gene clusters, or to elucidate biosynthetic pathways of known metabolites. I also use those genetic tools to elucidate genetic mechanisms regulating pathogenicity in fungi and to identify and characterize virulence factors. And lastly, I also use genetic tools to elucidate microbial host interactions or microbe-microbe interaction within a microbiome setting. But here I should mention that all these applications are well linked together, as a lot of fungal metabolites have been known to be virulence factors and play key roles in regulating microbiome dynamics. So advancing all those fundamental knowledge related to metabolism, pathogenicity, and microbiome organizations are so critical and constitute the basis for the development and implementation of novel genetic tools such as gene drive, which is the main focus of my talk today, to control fungal infections and diseases at the applied level. So I will start with a brief introduction of the fungal kingdom. So fungi are often overlooked in nature, although the role in the environment and in life is undeniable. So to mention a few, fungi play important roles in ecosystems by being the Earth's best degraders of organic matters, and also some of fungal species, especially those that are living in a mutual symbiotic association with plant roots, like mycorrhizal fungi, are shown to provide stable stocks of carbon and deliver nutrients essential for plant growth. But besides those ecological roles, fungi also play important roles in industry, either for food manufacturing or in pharmaceutical industries, like for drug discovery. As I mentioned, a lot of those fungi produced important secondary metabolites that have bioactive properties. But besides those beneficial function that fungi might have, some invasive fungal species can be real threats by causing life-threatening diseases such as aspergillosis or candidosis. Those are like the most known maybe fungal diseases. Or they can cause deleterious effects on ecosystems and significant reduction of agricultural productivity. And here I'm referring to a fungal plant pathogen. That's going to be the main focus of my talk, as I'm going to be focusing on a fungal plant pathogen called Sphaerulina musiva. So considering this double-edged role that fungi can play in life, it is critical to discover and characterize new fungal species, understand their biological roles and function, and get access to their genome information to, most importantly, develop genetic engineering tools to either improve the implementation and utilization of those benefits on the environment, industries, or to reduce the risk and negative impacts of fungal pathogens on humans, animals, ecosystem, and agriculture. So in my talk today, I'll be focusing on the pathosystems Sphaerulina musiva and Populus that we are working within the secure ecosystem and engineering and design SFA. So poplar trees are used across North America for several industrial purposes as potential source of bioenergy, biofuel, as well as for fiber production. But besides their industrial importance, poplar has essential function in agroforestry as they sequester CO2 from the atmosphere, and they require less water than other cultivations like corn, for example, and can reduce soil erosion and desertification. The genus Populus are also used, and we use it here at ORNL as a model organisms for the study of natural product discoveries, as well as for a plant microbial interaction. The main limitation to using Populus for fiber, biomass, and bioenergy in North America are the fungal diseases, such as the septoria leaf spot. So septoria has been the old name of Sphaerulina musiva. So that's why we call the disease septoria leaf spots and stem cankers that I'm showing in the images on my slide here, that are caused by the ascomycete fungal pathogen Sphaerulina musiva. So spirulina was first discovered back in 1884 on resistant Populus genotypes called Populus deltoides in New York State. So although it's original native for Eastern North America, it was only recently introduced and established into the Pacific Northwest of North America, resulting in deleterious effects on susceptible or what we consider naive Populus genotypes, such as Populus trichocarpa or its interspecific hybrids. And this had a greatest industrial impact due to the fact that a single infection can kill a whole tree. In terms of disease management to date, there has been a great amount of research that has been conducted on the plant side, either by creating mutants that have showed increased resistance to S. musiva or by identifying and planting resistant genotypes. There also has been other management measures to prevent the spread of this fungus and reduce its impact on ecosystem, mainly based on biological control, like the study in 2003, where they sprayed Streptomyces trained in the field to suppress leaf spot diseases. And in another recent study, they used Bacillus bilicensis strains. They were shown to have antagonistic activity against S. musiva. However, the use of these biological control approaches might be limited by the longevity or the activity of these bacterial strains in plantations. To my knowledge, there has been no genetic mitigation approaches developed on the pathogen side, and this is mainly due to the unavailability of effective transformation system to genetically modify this fungus, but also due to the limited knowledge on the molecular mechanisms regulating the establishment and pathogenesis of this invasive fungal species. So within the Secure Ecosystem Engineering and Design SFA at ORNL, we are focusing on developing genetic engineering tools to alter the outcome of microbial invasions by targeting genetic determinants demonstrated to facilitate the spread of this pathogen in vulnerable Populus ecosystems. So here's the overall approach we are following towards this goal. And later in my presentation, I'll be going more in details to every steps within this approach. So first, I worked on developing a CRISPR-Cas9 transformation system to be able to genetically manipulate this fungus. And we are currently working towards identifying genetic targets that we're gonna be using for the establishment of the gene drive system. So all those genetic targets that we are identifying either through transcriptomic or genomic resources, they're gonna be evaluated as potential gene-derived targets through knockouts and plant phenotyping assays. And all those efforts will be later leveraged towards the establishment of the safeguarding or split CRISPR-Cas9 gene drive. The first and only, to my knowledge, CRISPR-Cas9 gene drive system developed before on fungi has been developed on a yeast species called Candida albicans, which is a human pathogen. To my knowledge, no gene drive system has been developed so far on a filamentous fungus, and more precisely no CRISPR system has been developed on a fungal plant pathogen. So if we're successful, it will be the first CRISPR-Cas9 gene drive system on a filamentous fungal plant pathogen. So how does the CRISPR-Cas9 gene drive system works? For those of you who are not familiar with the system, it consists on linking an altered copy of a targeted gene to a CRISPR-Cas9 gene drive components, which are the guide RNA and the Cas9 endonuclease. So during sexual reproduction, the guide RNA will guide the Cas9 to the target site of the wild-type chromosome, as showing on my schematic on the top of my slide, to generate a double-strand DNA break at the target site. The double-strand DNA break will be later repaired through homology-directed repair using the drive-containing chromosome as a template. This will result in the guide RNA and the Cas9 drive and the altered gene being copied into the homologous chromosome, guaranteeing that it gets passed at higher rates, with nearly 100% of the offspring inherit the altered gene compared to the normal Mendelian inheritance. So as a critical first step towards the development of the system in Sphaerulina musiva, I developed the first CRISPR-Cas9 transformation system to genetically modify the species. This system consists on generating protoplasts from Sphaerulina musiva spores or germlings, and use those protoplasts for the co-delivery of the CRISPR-Cas9 and guide RNA ribonucleoprotein complex, along with a donor DNA. The ribonucleoprotein complex, or called for short RNP, consists of the Cas9 endonuclease and the guide RNA that target a specific locus. The RNP will generate a DNA double-strand DNA break that will be repaired through either homology-directed repair if I'm using a donor DNA that will contain a selectable marker. In most of the cases, the selectable markers that were used in those types of designs are antibiotic-resistant genes that will be used for the screening of our transformants. And if we are targeting a gene that allowed an easy phenotypical screening or auxotrophic screening, there will be no need for a selectable marker to be used. And therefore, the repair of the DNA double-strand break will occur through non-homologous and joining repair, creating different insertions or deletions at the cut sites, or what we call indels, resulting in an unfunctional gene. So for the validation of this transformation system, I first targeted the PRG gene that will allow an easy distinctive auxotrophy screening. This gene encodes for an enzyme in the uracil and uridine pathway. So when this gene is successfully knocked out or deleted, this gene will not be able to grow in the absence of those two amino acids. So I used the two web servers called CRISPR and RNA Fold in order to design a guide RNA to target specifically a conserved domain in the PRG sequence, taking into consideration essential criteria such as specificity, efficiency, that we want them the closest to 100, and off targets of the guide RNA, and of course, the lowest off target, the best for the guide RNA design. So after designing the guide RNA and doing the transformation system, I got a large number of transformants, of which I randomly picked 20 that I conducted PCR and sequencing around the guide RNA cut sites to verify that the cut has occurred, and we saw some insertion and deletions by mapping the sequences of the transformants to the wild type. So the insertion and deletion, as I mentioned earlier, they happened randomly, resulting in an unfunctional gene, and that's why we were able to screen those transformants on plates. The system showed 85% efficiency in transforming Sphaerulina musiva. So after developing the CRISPR-Cas9 transformation system, and in order to guide engineering of the gene drive for more sustainable and productive ecosystem, the second step consisted on identifying genetic determinants that are implicated in the establishment, spread, and pathogenicity of this invasive species. So here's a schematic representation of the overall strategies we are following to achieve this goal. So first, we are developing, with the help of the computational team at ORNL, a S-musiva pangenome representing 122 isolates of this fungus collected across the US. And we are also generating a genome-wide association study of what we call GWAS resources. Also, in collaboration with Jared Lebaldis at Oregon State University, we are conducting comprehensive phenotyping, transcriptomic, and metabolomic analysis on these isolates in order to advance the genotype to phenotype discoveries, and which would allow us to correlate any variations that we see in virulence on those species or on those isolates with both the SNPs or absence and presence of genes in the Sphaerulina musiva pangenome. All the genes that we were able to identify from the transcriptomic analysis or the genomic resources, their function will be later validated through CRISPR-Cas9 knockouts using the transformation system that I just talked about, and through plant phenotyping assays to confirm that they are truly implicated in virulence. In the first efforts to identify the genes governing stem canker formation in Sphaerulina musiva, we have conducted a transcriptomic analysis at 2 and 3 weeks post-inoculations of stem with Sphaerulina musiva wild-type spores. These analyses have allowed us to identify several genes that were shown to be highly expressed during canker formation at the two tested time points, the 2 and 3 weeks post-inoculations. Among the genes that we're able to identify from those transcriptomic analysis is the effector gene called ECP2. And this specific gene was reported in other pathosystem, like in Valsamali, to be implicated in fungal pathogenesis. So this gene, as I mentioned, it was highly upregulated during infection and stem canker formation. And when we did an infiltration assays on leaves, it was shown to invoke hypersensitive response on susceptible genotypes. So to validate the role of this effector in virulence, I used the developed transformation system to knock out this gene and confirm by nanopore sequencing the absence of Cas9 off-target effects in my mutants. Later, I used one of those mutants to perform pathogenicity assays on detached Populus leaves. So this was done by creating artificially small lesions on the surface of the leaves and inoculating with the same amount of spores from both the wild type and the ECP2 mutants. So the first data collected on Populus hybrid genotypes showed a significant reduction of leaf spots area in the delta ECP2 mutants compared to the wild type. But since ECP2 was identified during stem canker formation, we are currently conducting comprehensive analysis on different stems of different Populus genotypes to further validate the role of ECP2 in Sphaerulina missiva's virulence or pathogenicity. Also, to identify genetic determinants regulating Sphaerulina missiva establishment in Populus soil microbiome, I recently conducted another transcriptomic experiments through which I was able to optimize an enclosed system that mimicked the natural conditions of presence of this fungus in the soil, like in the wild. So for that, we used soil that was collected from a Populus field in Oregon that is known to not have septoria in this field. And we used two conditions of soil: autoclave soil that has a reduced microbial community and non-autoclave soil that has the native microbial community from the field. I also used two different Sphaerulina missiva isolates that were shown to have different degrees of virulence. So one of them was shown in previous study to be highly virulent, and another strain that was shown to be less virulent. So this is the system that I used, and after 10 and 25 days post-inoculations, I collected soil and I did RNA extractions, and we sent those samples for transcriptomic to look for any differences in the presence and absence of microbial community. So after filtering the transcriptomic data by significant p-value and log-fold change, I was able to identify 16 genes that were significantly differentially expressed in the presence versus absence of microbial community. Of those 69 genes, 14 were shared between both isolates, the low and the high virulent isolates of Septoria. Interestingly, among the upregulated genes in the absence of microbial communities, I identified some genes that encodes for enzymes previously reported to be implicated in fungal pathogenesis, such as phospholipase, acetyl-CoA ligase, and adenosyl homocysteine, and enzymes reported to be implicated in Postoria and biotrophic colonization. So the upregulation of these enzymes is a potential indication of the fungus getting ready for host invasions in the absence of microbial competition. Among the genes upregulated in the presence of community, I identified a gene encoding for senescence-associated protein, which is not surprising, as senescence is a protection mechanism triggered by conditions that impose stress, like in our case, the presence of either other microbial competitors. So I'm currently following up with knockout studies on these genes to further validate their role or their functions in the establishment of Sphaerulina in Populus soil microbiome and invasion, as we did for the ECP2 effector gene. So aside from the genetic targets that we're able to identify through transcriptomic, we also used the population genomic resources and identified a strong correlation between stem canker formation in the susceptible Populus trichocarpa genotype and a selected biosynthetic gene cluster encoded in Sphaerulina missiva genome. So this biosynthetic gene cluster is predicted to encode the genes responsible for the regulation and synthesis of a mycotoxins called Atpenin A5. So this biosynthetic gene cluster, as indicated with my red bar, was found to be absent among Sphaerulina musiva isolates collected from the Pacific Northwest region that do not form stem cankers in the tested Populus trichocarpa genotypes. So here's a figure showing the different domain in the PKS and RPS backbone gene of the Atpenin cluster, showing the absence of certain essential domain in some of the British Columbia isolates, which indicates that the gene is unfunctional and therefore an impairment of Atpenin production in these isolates that were confirmed experimentally in our lab. So Cole Sawyer, who's a graduate student that I'm mentoring on the fungal transformation part, is currently doing or generating knockout strains of these genes in the Minnesota strain to further assess the role of this biosynthetic gene cluster in stem canker formation. That can also be potentially another gene target that we can use later on for the development of the CRISPR-Cas9 gene drive. So after optimizing the CRISPR-Cas9 transformation system, identifying some potential gene targets that we are currently validating their function in virulence, I started working on implementing the CRISPR-Cas9 gene drive system by targeting the effector gene ECP2 that we confirmed that is potentially implicated in virulence. So I already briefly explained how the CRISPR-Cas9 gene drive system works. So here's a comparison between a normal Mendelian inheritance and a gene drive inheritance. So through a normal Mendelian inheritance, an altered gene has a 50% chance of being passed to the descendant. So when inherited in this way, the altered gene will be diluted over several generations and will not be spread to many individuals in the population. However, if the altered gene is linked to a gene drive, as shown in the schematic on the upper right, the CRISPR guide RNA will guide the Cas9 to the target site of the wild type chromosome to create a double-strand DNA break at the target site. And this will be repaired by homology-directed repair using the gene drive components as a template. As a result, the guide RNA, the Cas9 drive allele, and the altered gene will be copied into the homologous chromosome. Therefore, in that case, nearly all the descendants will inherit the drive allele, and it will spread through the population. So here we have to, I have to mention that when both elements, the Cas9 and the guide RNA, are present together or at the same locus in the genome, we call that an active or a full gene drive. And then this strategy can be so effective that the alleles can spread even if they are conferring disadvantageous traits. In our case, we are working on developing what we call a safeguarding or split gene drive, where we're going to be inserting the Cas9 and the guide RNA separately, so on two different genetic elements, and they will be inserted separately at two different loci in the genome. So here's the experimental approach that I'm using to engineer the split gene drive strain. So I'll be starting with the wild type strain that for which I will be doing first a transformation to introduce the first element or component of the gene drive, which is the guide RNA targeting the ECP2 locus. The transformed strain will later be used for another transformation, of course, after genetic confirmation that the first transformation occurred correctly at the right locus and there was no bugs during the homologous recombination events. It will be used to introduce the second component of the drive element, which is the Cas9 at the pure G locus, to get our drive strain having both components inserted at different loci. So using the same web service, CRISPR and RNA-fold that I talked about it earlier in my presentation, I designed a guide RNA on the HCE2 conserved domain of the ECP2 protein. So we designed the guide RNA on conserved domain to make sure that when the cut and the repair is occurring, the gene is losing its function. And that's why we target conserved domain. And also, of course, I looked at criteria such as specificity and efficiency that are 100% and zero off targets of the guide RNA. Later, I looked at the polymorphism across the Sphaerulina population that we have in our lab at the guide RNA target location. As we can see in the table, the percentage of compatible strain is high in a lot of isolates collected from different states, which means that there is less polymorphism at the guide RNA target site in these strains, which is something that we don't actually want. We want to have certain types of polymorphism that later will allow us to use the strains for the confirmation of the success of the drive. Therefore, for the design of the gene drive, I have focused on the Minnesota strains for many reasons. First, they showed a higher polymorphism at the ECP2 guide RNA target location. They are well studied in previous studies, so there's a lot of publications on those specific isolates and likely to be easy to manipulate and to develop a mating assays. And also because those strains have showed a high level of virulence compared to isolates collected from other states. So here's the Plasmid Map that I designed for. So the first plasmid include the design guide RNA surrounded by self-cleaving ribozymes, which mediate cleavage of the RNA, which are the hammerhead and the HDV ribozymes that will mediate a cleavage of the RNA at the 5' and the 3' ends to generate transcripts. This contract also includes the HPH gene encoding for hygromycin resistance that will allow the screening of our mutants on hygromycin supplemented media. The other backbone that I designed includes the Cas9 encoding gene under the PDC promoters and terminator. So for this transformation, I used, as I said, one of the Minnesota strain, wild type strains called Minnesota MN5, and I used a linearized plasmid that I just talked about carrying the guide RNA. Following the same transformation protocol that I have optimized, I did the transformation of the Sphaerulina musiva strain MN5, and I used CRISPR-Cas9 ribonucleoprotein complex and donor DNA to have a homology-directed repair at the ECP2 cut site. So I used the linearized vector that I just showed as donor DNA, which has homology arms that will allow the recognition at the ECP2 locus. The selection has been done on plates supplemented with hygromycin that has been used as selectable marker. From this transformation, I got a large number of transformants, and I always picked randomly 20, on which I performed a second screening on plates supplemented with hygromycin to eliminate all the ones that might be false positive. And only 14 transformants were able to grow on the hygromycin plates. And of those transformants, I did 2 PCR reactions to validate the insertion of my construct at the right locus. So the first one using the 5' forward and reverse primers, and on the other side, the 3' side, to validate the insertion of my construct. So here is the PCR results showing that almost all the transformants except one did not have the insertion of the construct at the right locus. The others all have it at the right locus by giving me the expected bends at the 5' and the 3' flanks. Later, I also performed a whole genome nanopore sequencing on some of the positive transformants that we found by PCR. And through mapping to the wild type genome, I was able to confirm that I have a single insertion of the guide RNA construct, because we don't want to have multiple insertions in other loci in the genome. We want just a single insertion at the ECP2 locus. And I was able to confirm through whole genome sequencing that there were no SNPs detected due to the Cas9 off targets. So after confirming that the first component of the gene drive system has been inserted correctly at the targeted locus, I'm currently working on I performed the second transformation, but I'm working on validating the mutants that I collected from the second transformation using the plasmid that contained the Cas9 construct targeting the pure G locus. So I'm currently, as I mentioned, screening those transformants from the second transformation and hoping that I was able to engineer the drive strain that has both the guide RNA and the Cas9 inserted correctly and functional at two different loci. So later also I'll be doing a whole genome sequencing on those mutants to check for any off-target effects of the Cas9. So in parallel of the genetic engineering of the drive strain, I'm also currently working on optimizing the mating experiments that I will be using later on for the confirmation of the gene drive success. So here are the strains that I picked to optimize the mating experiments. Of course, they have different mating types than my MN5 drive strain. So I picked the MN5, the Minnesota strain 10, that is supposed to be cut by the guide RNA that I designed and also have a different mating type than the MN5, and Minnesota strain 21 that should not be cut by the guide RNA and it can be used as a negative control. And in that case, we should be expecting a failure of the drive system to work in this mating. So developing the mating assay has been quite challenging compared to other fungal systems like Fusarium, for example, that they can mate on agar plates. So we tried the in vitro plate system for Sphaerulina musiva, but so far I wasn't successful in getting it to work. So I'm working currently on developing a mating assays that mimic the natural conditions of sexual reproductions of the species in the field. So for that, I would explain, to further explain that, I have a schematic representation of the Sphaerulina life cycle. So you'll be able to understand when is the sexual reproduction happening in this life cycle. So in spring, the primary infection occurs by means of ascospores that will penetrate through to the leaves through the stomata, which are the natural opening of the leaves. So the initial lesions will appear 3 to 4 weeks after bud burst on the leaves of the lower branches. Later during summer, the pycnidia, which are the asexual fruiting bodies, will be formed on the lesions on the leaf surfaces and in cankers. Canidia released from these fruiting bodies will be dispersed later on by the rain and the wind, causing additional secondary infections on leaves and stems by penetrating through the petioles, the lenticels, the stomata, or even through wounding sites. So here's when the sexual stage will begin in fall with the formation of spermagonia on fallen leaves. So the leaves that fell from the trees and that are naturally senescing on the soil. In these leaves, numerous hyaline rod-shaped spermatia, which will be produced, and they will give rise to fruiting bodies called pseudothecia. So although the mechanism is not clear, it has been hypothesized that pseudothecia could possibly act as survival structure for the fungus during the winter. And then the cycle will restart again with the asexual spores. So to summarize, the sexual stage, as I just talked about, is known to occur during the fall and winter on falling and naturally senescing leaves. So here's a schematic representation of the mating assay that I'm developing in the lab, and a similar assay. So we were inspired by a similar mating assay that has been successfully conducted on Zymoseptoria, which is a closely related species to Sphaerulina musiva or Septoria musiva. So I'm feeling pretty optimistic that I will be able to get this mating assay to work in the lab. So to mimic the natural conditions of sexual reproduction, I will be performing co-inoculations with pairs of compatible parental strains and single inoculations of each parent to evaluate the crossing possibilities by spraying adult Populus plant with equal suspensions of conidiospores from the two parental strains. The plants will be stored in an enclosed system that will allow the gas exchange because we don't want to kill the plants, but it will avoid cross-contaminations in gross chambers under regulated temperatures and humidity levels. And we're going to be having a light and dark photoperiod to allow those plants to follow their natural development cycle and senescence. Later, the senesced leaves that will be naturally falling in the bags will be collected under sterile conditions and stored for additional months in plastic bags to allow the sexual reproduction to happen. Later, at the end of the incubation period, I will be collecting several offspring strains, and they will be genotyped using the random amplification of polymorphic DNA, or what we call a rapid PCR method, to check the parenthood by comparing the genotypic profiles of the offspring to the parental strains that I use for those assays. So to conclude on the advancement that I achieved so far in the development of the CRISPR-Cas9 gene drive system in Sphaerulina musiva, I was able to identify genetic associations for several important establishment and pathogenesis traits that are in the process of validation through CRISPR-Cas9 knockouts and pathogenicity experiments in planta. As I showed in the presentation, we're able to confirm the role of ECP2, which was one of our first targets to be implicated in virulence. I was also able to successfully develop the first protoplasm-mediated transformation system in Sphaerulina musiva using a protein-based version of the CRISPR-Cas9 genome editing system. And I'm currently working on leveraging from those efforts to develop the establishment of the first synthetic CRISPR-Cas9 gene drive on a plant pathogenic fungus. At the end, I would like to acknowledge all the team that is working alongside with me on this project. I would also like to thank at ORNL as well as at Oregon State University. I also would like to thank the Secure Ecosystem Engineering and Design SFA for funding this project. I would like to thank you all for attending the webinar and listening to my presentation, and I'm ready to take any questions that you might have.
Great, thanks a lot, Joanna.
Thank you.
Really interesting. And let me just get myself organized here just for a second. So let me encourage people to ask questions, and let me remind you that there are actually now two ways you can ask questions. And one, you can chat those in. And again, I encourage you to chat in any kind of questions that are coming to your mind. I'm sure there was a lot of content there, that some of it highly technical and some of it less so. So feel free to query Joanna on anything you'd like. I'm sure she'd be more than happy to talk about her work some more. Now, you can do that by chatting, or you can also, if you raise your hand, we will recognize you and we can unmute you, and you can ask your question orally. So I'm just going to give some people time to ask some questions here. I know we have a couple already, but I want to get a couple of mine in first, just so I don't lose chance. So first of all, really interesting, and the whole development pathway that you've explained, and the technical nature of it, and trying to develop these things anew, basically de novo, is really exciting, and you're really obviously making lots of progress and having a lot of success. I had a couple of questions. I mean, you towards the end, you got to the life cycle of the fungus, which was really interesting, and of course there's sexual and asexual stages to these things. And so thinking about some of the questions that come to my mind have to do with how you envision such a control mechanism being implemented in the field, for example, how the asexual stages might impact dissemination and spread, those kind of questions. So I guess the main question I'd like to get to is, it seems to be a soil bacteria. So how would you think about this being used in a way that for control?
Sure. Yeah, a lot of people I presented it at a conference like a week ago, and a lot of people have the same concern, like, are you going to be able to release those GMOs organisms into the wild?
Yeah, so my question, my concern wasn't as much the GMO part. Mine was really more about the biology, about the implementation of the strategy, for doing that. But you can
Yeah.
speak to both issues for sure.
Sure, yeah. So for now, most of the experiments are going to be done in growth chambers. So we have established growth chambers and established greenhouse, like kind of like a walk-in growth chambers. We're going to be using for the confirmation of the drive on plants. We are like too early before getting it to like establish in the field. For now, we don't have any permit on releasing those organisms or testing them in the field. So as a first step, it's more like a proof of concept of getting this system to work in a fungal plant pathogen, because as I mentioned earlier in my presentation, it hasn't been done before. So yeah, it's more like as a proof of concept more than like the actual control, because there's a lot of steps to get to before getting it to test it in the wild or in the field. I'm not sure if I answered your question.
Yeah, it's okay. Yeah, it's fine. There are a number of questions coming into the chat, so I don't want to linger on my comment, and I want to get to some others. First of all, there was a comment from Zizou Kome, I believe, in Benin. I'm going to let you read that. It's not really a question, but it's a comment about your work and some interests that they have. So I'll leave that. But I wanted to bring that your attention, and we'll capture that too, and be able to send that to you as well. So Zizou, thanks for the comment, and Joanna, we'll get back to you.
Sure.
Victor, so I think it's Victor Lopez. Yes, it is. Delamo says, are the Cas9 and guide RNAs, the gene drive elements, linked? And the comment is that this would be good to track donor versus receiver chromosomes. So yeah, go ahead.
No, the guide RNA and the Cas9 are not linked. As I mentioned, they are inserted at two different loci. So we use the ribozymes to generate the transcript of the guide RNA that will go and bind on the wild type locus during the sexual reproduction, because the spores are, I didn't maybe mention that during my presentation. So the spores are haploid. So during the sexual reproduction, both haploids will kind of like meet, and therefore the guide RNA on the drive strain will direct the Cas9 on the wild type chromosome to create a cut. So no, in the design, the guide RNA and the Cas9 are not inserted. That's why we call it a split gene drive. And so as I mentioned, it was developed first in Candida albicans, and we're currently following kind of the same design they had for the split gene drive in Candida. And yeah, hopefully we will be able to get it to work in Sphaerulina.
Great, thanks. Let me just repeat my encouragement to the audience to ask questions, and also if you'd like to raise your hand, you can cut the line of chat questions, and I'll recognize you right away. So yeah, I guess just following up on that. So the idea of using a split drive here is being the interest in split drive in this case is what? Why split drive and not
it's like safer than the full or the active gene drive, because when the guide RNA and the Cas9 are linked together on the same loci, they are so active. So having both elements separate will be able to control in case like we got disadvantaged traits. Like here we are targeting a virulence factor. We are hoping to get less virulent strains through the gene drive, but you know, like in genetics and in microorganisms, we might get some unexpected results. So therefore, in case something happened or this drive gave us a disadvantaged traits, we don't want to be generating a more aggressive or virulent strains. Therefore, we designed it this way as a way of control than having an active gene drive spreading through the populations.
Yeah, good. Yeah, I seen Wayne Landis has his hand up. I'm gonna, let's see, can we unmute Dr. Landis and let him ask his question? Dr. Landis, are you there? Yeah, just if you can unmute yourself, perhaps. Jasmine, can you see Dr. Landis and the participants? Okay. Well, his hand is down now. I don't know. Maybe put his hand down. Okay. He
Keep
has a question.
unmuting myself.
Ah, there you go.
Oh, good.
Wayne, good to talk to you again, and glad to
Yes.
see you at the webinar today, and really interested in hearing some of your comments and questions.
Thank you very much. It's also, I put my question in the chat.
Yeah.
One of the issues with gene drives is the development of resistance. And of course, with microorganisms or things that reproduce quickly, even low probabilities, the probability of resistance becomes 1 given enough time. So that will prevent the movement of the drive, perhaps. And have you investigated what potential resistant pathways would be?
We are still, yeah, thank you for your comment. It's a very important comment. And since we are still in a very early stage of the development, we didn't investigate if that's something that's going to be happening and to what extent it might be happening. But it's something that we will definitely take more into considerations when we progress further in the development. But we are in the very first steps or stages of the development.
Yeah, I understand that. But I also worked with, I was one of the panelists for the National Academy Gene Drives on the Horizon. And the risk assessment part, I think that's important to be considered early on. Because there is a huge, I mean, as you mentioned, these are early days, but also you've already spent a huge amount of investment, both in time and money. And I guess I would suggest that that consideration would be important.
Sure, yeah.
But this is really cool. This is really cool stuff.
Sure, thank you for your comment and for your great suggestions. And yeah, we'll definitely take that into consideration.
Great, thanks a lot, Wayne. Actually, there was a question that the next one on the chat is related to this resistance, and it's from Simon Lilico. And how concerned are you about resistance allele arising due to non-homologous end joining? And is there anything you can do to bias repair in favor of directed
so repair? So when we did the first steps of transformation in the PRG and the ECP2 strains, we looked for the percentage of non-homologous end joining occurring. It's not so high in this fungus compared to other fungi that I worked with, but we are also considering on generating a Q70 strain, which is a strain once we create a deletion of the Q70, we will reduce non-homologous end joining. So yeah, that's something also that I'm currently working on generating, and we're probably gonna be using it for the development of the drive as well, to kind of think about the potential of reduction of non-homologous end joining in the favor of homology-directed repair.
Yeah, I'm just going to slip in a question here. You mentioned HC2 as a target for your insertion of the guide RNA, or the target for the it was a target for the guide RNA. Could you say something? What is that gene? Why?
So the HTE2 is a conserved domain in the ECP2 virulence or effector genes. So conserved domain will give the function of the gene. So they are part of certain families that give the function to the protein. In my design of guide RNA, I always try to target conserved domain. So in case non-homologous end joining were occurring, for example, in the case of the PRG, sometimes if there's small SNPs, the function the gene won't be very affected, and therefore it will stay functional. However, by targeting a conserved domain, we're making sure that we are removing the function of the genes, because we are targeting a very important, let's say, or a very important domain in the protein.
Yeah. Good. So Wayne has already asked this question, but there was an element of his question that I'd like to bring up and had to do with any modeling that you've done on this system that might be informative to questions about dissemination, how efficient it might be in the wild, risk, and things like that.
Yeah, I see a comment, I think, about do you envision the drive? I'm not sure what comment are you exactly talking about.
Well, it was one of the questions that Wayne had in his comment was, has any population genetics modeling been done yet to evaluate the efficacy of the method? So, so I'm just sort of picking up on whether or not there's been any modeling done in your group for the system that might give you ideas about how this might move through a population of these fungi.
We have not done that yet, but yeah, it's something that we're gonna be using.
Yeah,
for testing.
Okay, good. I'm going to go to Ethan Beer's question. It says, how do you envision using the gene drive system you're developing to get the system into the wild type virulent strain? And how might you try to overcome the problem of cleavage resistance alleles in the target site? So yeah, so we've talked a little bit about the resistance issue, but the first part of the question is something would be interesting to hear your thoughts on.
I would like to read the comment again.
Sure, it's Dr. Ethan Beer right down there. It's how do you envision using the drive system to develop, you're developing to get into, how do you get that into the wild virulent strains?
Into the wild virulent strain. I'm not sure if I understood the question well. I will answer the way I understood it. So the wild type would have the ECP2 functional copy. So the guide RNA that I designed will target a specific locus on the wild type chromosome during the sexual reproduction. It will generate the cut, and to repair the cut, we're hoping that it will be repaired by homology-directed repair using the template that we have on our drive strain to repair the DNA double-strand break and transferring the gene drive components into the wild type. And therefore, the wild type would have now the gene drive components, and it's going to be acting as a gene drive again. Did I answer the question?
I don't know. I can ask Ethan. Ethan, if you want to raise your hand, go ahead and do it. We'll recognize. Okay, there you are.
I think he's also asking another question regarding why we use.
Ethan, Jasmine, did you unmute Ethan? Ethan, go ahead and try to unmute yourself and go ahead and talk. Jasmine, could you unmute Dr. Beer? Here we go. Go ahead, Ethan. Thanks a lot. Hi, Ethan.
Very interesting talk. No, I guess the question I had is just a very simple one, which is, if you have a strain that you've developed, how do you get that strain, given all the complexities of the soil? I don't know if these organisms are kind of unitary organisms in the soil or if they already exist as communities. But how do you get them to mate essentially with the target organism and do so in large enough numbers so that you have an impact? And of course, as some of the prior questions were asking, how do you then also deal with the fact that the ones that get NHEJ mutations, they're the ones that are going to still potentially, especially if they're functional NHEJs, they're the ones that are going to be able to outcompete in terms of making the disease. And so how do you, deal with that problem of getting your drive into a substantial proportion of the others? Are there things like in bacteria, you have biofilms where you can get, very efficient transfer of a genetic element from one organism, one cell to many others? Are there things like that going on that you could exploit, or how do you imagine just using it? I'm just trying to understand exactly what the product would be and how you might intend using it. I guess that was the overall question.
Now I understand. So that's why we were hoping originally to have it to work in an in vitro system, like on plates, like what they do for Fusarium, for example. But unfortunately, it didn't work. That's why we had to move to a more complex system. And as I said, we were inspired by the mating assay that they used on Zymoseptoria, where they did it on plants. And so in that cases that we're going to be doing on plants, so the spraying will be happening on plants, not in soil. But for that system, we're thinking of using a soil with a reduced community. So we're not going to be introducing any more fungus or microbes other than the 2 strains or parental strain that we want to be using. And therefore, we're going to be observing the sexual structures that are going to be occurring. Usually that's how they do it in Fusarium, but they observe those sexual structures, they collect them, and they do a screening, the rapid PCR, to make sure that they're coming from the 2 parents that they used. We're also thinking of doing whole genome sequencing later on to confirm those descendants coming that from the 2 parental strains that we introduced to the system.
Good. Okay. I see. Ethan.
I think he had another questions regarding why we're using the hammerhead.
We'll get to the one on the hammerhead in a minute or two. I just wanted to. So it seems to me that with a spore forming system like this, that thinking down the road, if you had a product that you wanted to introduce into the environment, that spreading spores would be probably the modality to do that. I would imagine. But I guess the questions are mating compatibilities between the transgenic strain, the derived containing strains, and the wild strains, which I think is, yeah. So Ethan, go ahead again.
Okay, so I guess, again, I was kind of thinking more in the long term of implementation, like what would the strategy be? Would you take the gene drive system and spray the tree leaves with it? Or would you wait till the winter where the leaves have fallen and then try to get it into the soil mat? What would be the target, and how would you anticipate having efficient transfer of the genetic information in the context of the wild, where, the trees are infected in places where there's wind and precipitation and all the stuff that happens in the wild? What would be the approach that you would take? Would you do it multiple times during the year when they're on the leaves? Would you, I'm just trying to understand the, with a mosquito, you take a mosquito, you jump it out into the wild if they let you do it. And then the mosquito finds, the male finds a female if it works. And then, that's how it goes. But in this case, how would you do that exactly? Is mating required, for example? Are these, is that 100% true? Is this a completely sexual dependent organism?
So it can multiply through asexual and sexual reproduction, but for the gene drive, we believe that it needs the sexual stage to be able to spread. And the way we envision it, if we were able to get to the field applications, which we are still so far behind on doing that, we were thinking of spraying on the tree. So it's something like when they use, for example, biocontrol of, as I mentioned, the streptomyces strains or the bacillus strains that they use as a biocontrol against Sphaerulina. We were thinking of using the spray method the same way to introduce the gene drive strain, and the leaves will eventually gonna be spreading because they're gonna be forming more ascospores, which are the asexual spores and the asexual structures, and they're gonna be even more sporulating. And with the wind and the rain, that's gonna be transferring to the whole plants through natural openings on the stems, on the leaves, and other openings or wounding on the plants. So that's where we're hoping the transmission of the spores were going to be happening from the gene drive.
Do you know whether there's any system that exists for conjugal transfer from bacteria into fungus? There are ways that there are systems of conjugal transfer from bacteria into mammalian cells. So I'm just wondering if you could use some of those same tools that you're using to control the fungus to also use them as vehicles for conjugal transfer of a plasmid that might contain your drive element that could get it in as well, so that those they didn't kill, maybe they transformed and then reduced the virulence. Is there a possibility of using that kind of strategy?
Like you mean like some kind of like of horizontal gene transfer between bacteria and fungi, or vice versa?
Exactly.
It's a possibility. We didn't actually think about it. It's a good point that you mentioned. We didn't think about it earlier. We know like those events happen, and there's like so many evidence of genes like being transferred from bacteria to fungi, or like a gene that hasn't been described in fungi before and happens to be coming from other organisms or other kingdoms. It is a possibility. We didn't think about it earlier.
Yes, it does. Sorry, I'm just going to bump in here for a second, but you can stay on if you want to more comments. I'm welcome the discussion. But does mating take place in when the fungus is in the leaves?
On fallen leaves. So actually, even the life cycle that I mentioned, some part of it is hypothetical. There's no like actual proof, but from what people saw and published, they saw the sexual structures occurring on senesced leaves, not on leaves that are already still on the trees. That's why I said during the developing of the mating, we are trying to have the leaves going through their natural cycle of senescence. Because like you can even take leaves and spray those senesced leaves by the gene drive, or like the mating parents that you're hoping to get the mating assays on, but we wanted to do it as closely possible as the natural system that happens in the wild.
But in terms of trying to get the drive to spread through a population, you'll need to get it. It's got to be mating with the wild types that are out there. And that seems to be occurring in the soil, if I understood your life cycle.
On the leaves that are deposited on the soil, kind of, yeah.
But okay. So I guess an interesting biological question then, that really, in terms of knowing some of the details of the life cycle could be really
exactly. And we're actually working towards more elucidation of the life cycle. I have other colleagues in the lab working as well on elucidating more the life cycle to try to understand it more, because that will help eventually in the it's a very unknown pathosystem, let's say. There's a lot of stuff that we don't know about, and working on an unknown system has been like quite challenging, from like the non-presence of transformation system when I first started this project, or the unknown molecular mechanisms of how this fungus become virulent, what are the genetic determinants of virulence. So yeah, there's a lot of unknown that we are facing.
I just want to acknowledge that Zhang Jing Shen in the chat has had a question, but I think we've covered that. It had to do with the reproductive, sexual, asexual reproduction. So I just want to acknowledge that I see your question, but I think we've covered it. So I'm just going to move on. I'll get to Ethan's question about the hammerheads in a second. Also, Ivan Mugani had a question about, would the drive be applied to plant or the fungi? And I think our conversation in the last minute or two was getting at those questions. So I guess just want to acknowledge, Ivan, that I saw your chat question, and I think we covered it. So just to get down to, I guess, the some of the technicalities about this, the ribozyme. I had some questions about its function in your plasmid, and Ethan Beer has a specific question. I'll use that as the basis here. Why did you use a hammerhead for expressing the guide RNA instead of a Pol3 promoter such as U6 used in other gene drive systems?
We didn't have a preference for the hammerhead or the HDV. It's more like it has been used in the Candida albicans system. And since we're trying to use kind of like another closely related system, which is a fungal yeast stage, we thought that it might be a good choice to start with that. But also, like, we don't know if it's actually going to be working. So we have other options in case the hammerhead and the HDV don't work for our system. But yeah, we didn't have like like we picked them on preference. It's more like we were looking what has been done in Candida so far.
Yeah. So I guess I'm a little unclear about the function of the ribozyme in that construct. Was it actually to promote expression of the use of the guide RNA? Or
yeah,
it
will cleave the guide RNA on the 5' and 3' ends
okay,
to generate transcripts. Yeah.
Yeah. Okay. Yes, I see. Okay. Great. I don't see any more questions at the moment in the chat. I just wanted to get a clarifying question. You mentioned that when you were looking for strains, that polymorphism in the guide RNA site was a desirable thing, and I didn't quite understand why that was when you were talking about it.
Yeah, so I mentioned that it's more like a justification why we are focusing on the Minnesota strains. It would have been actually easy to work on the Tennessee strains since we're in Tennessee, and it will be maybe easier if you want to like perform some experiments maybe outside the lab. It would be more understandable to use the Tennessee strains. So regarding the polymorphism, when I designed the guide RNA, I mapped the guide RNA cut site throughout the population that we have, and I found that in isolates from other states, the guide RNA was very conserved. Therefore, if I want to get a negative control strain that should not be cut by the guide RNA, it's kind of like a negative control for the drive system, was hard to pick it from other isolates since it's so conserved. Therefore, in the Minnesota, I saw a lot of polymorphism at the guide RNA cut site, which allowed me to find strains to include as a negative control in the system. Because I want a strain to be able to be cut by the guide RNA, a strain that is not able to be cut by the guide RNA. And plus, the Minnesota strains are the most abundant strains. So we have like maybe 30 isolates from Minnesota, so we had a larger choice to create any or to use any strains in the mating assays.
Yeah, and again, if there's any lingering questions, ask them now, because we'll probably wrap things up here in a few minutes if there aren't any additional questions. So I did want to ask you about whether or not, if you know, some of the spore killing systems that exist in other fungi actually will function in your system. Of course, I would imagine you haven't tested them yet, but I'm just thinking about the interesting experiments that Don Gardner did with the Fusarium, where he took spore killing system from I don't know, Spotophyra, I don't know which fungus it was.
Neurospora, yeah.
Yeah, and he was able to put it into Fusarium into a virulence determining loci, and get it to drive, at least in the laboratory. So
yeah. I'm aware of a lot of like the spore killing system that are developed in mainly Fusarium, as you mentioned. I know like Anna Calvo's labs also working on identifying some of the selfish genetic elements that can be used for the spore killing system. At the beginning, we were debating between using a CRISPR-Cas9 gene drive or a spore killer system that it's more like a natural system that people are working towards developing a synthetic spore killing system. The only thing is about the spore killer system, since it's occurring in fungal structures that are not easy to isolate, the system is not very well understood, whereas for the CRISPR-Cas9, we know how it functions. Therefore, if we have to troubleshoot anything down the line, for example, we can test for the Cas9 ability to cut in the lab, in vitro. Whereas for the spore killing system, the system, or it's known as how it is working, but the details of it, if you want to do any troubleshooting, is not very well known yet, because as I said, it occurs in structures that are very hard to isolate from fungus. Therefore, we got to pick the CRISPR-Cas9 system.
Yep. Okay, good. Before we wrap up, I'm just going to mention that this is not the last webinar in this series, and that we actually have one more in this series that will take place in 2 weeks from today. It'll be Peter Atkinson from the University of California, Riverside. He'll talk about some work he's doing on drive development in some a group of insects which include a large number of insect pests, the hemipterans. So let me stop that and say, Joanna, thanks a lot for the webinar today. It was really interesting, and I generated some questions, and people are thinking about it, and so that was really, really good. And so thanks a lot.
Thank you so much for the invitation, and thank you all for attending the webinar. I was really very happy to present my work, although we are still in the early stages of the development, but I got a lot of good feedback, good ideas that I'm going to be for sure including in the thinking towards the development of the system, and a lot of very good comments from all the participants in the webinar. And
Great.
Thank you so much.
Good. Yeah. So we'll call it a successful day. So that's one of the things that we're hoping for. So thanks a lot, and thank you to the audience. Thanks for all the interactions from the participants in the audience. And with that, I'll say have a nice day, and hope to see you back in 2 weeks for the last webinar in this series. So thanks.
Thank you.